Hello, everyone, and welcome to our conversation with an update on the TELLOMAK clinical trials. I'm Susan Thornton, the CEO of the Cutaneous Lymphoma Foundation, and today we have two amazing and wonderful clinicians and researchers with us. Our conversation with Dr. Pierluigi Porcu, who joins us from the Sidney Kimmel Cancer Center here in Philadelphia, and Dr. Professor Martine Bagot, who joins us from the Hospital Saint Louis in Paris, where she's head of the Dermatology department. And as many of you know, both of our esteemed clinicians are experts in the cutaneous lymphoma space and have been doing a lot of research and work in our realm. So today we're specifically talking about the TELLOMAK trial, and welcome Dr. Porcu and Dr. Bagot. It's really lovely to have you both with us today.
Good morning, Susan. Thank you for having me on. It's great.
Good morning, Susan. It's a pleasure to be with you all today.
It's lovely to have our coast to coast across the Atlantic. So it's really, thank you for taking the time to have the conversation. And I thought we'd start with a little background because most patients don't get to see what happens with a therapeutic until either they participate in a clinical trial or it becomes available and it's a treatment that they can get from their physician. So I thought, Dr. Bagot, you really were the key investigator that found this particular marker that was predominant in cutaneous lymphoma patients. And I'd love for you to share with our patient community a little bit about that process, because that is the really important research that happens way, way, way before we even find a potential target to use as a therapeutic, way before even a phase 1 clinical trial. And most people in our community don't get a chance to hear that story. So I thought I would let you start off by giving us some background on how did you do this research? How long did it take? What were some of the ups and downs, and how did this all happen to get to the point where you could have a therapeutic target?
Thank you, Sue. There was quite a long time ago, approximately 20 years ago, and at that time, it was a time where scientists were exchanging antibodies and trying to define clusters of differentiation and try to find new targets. And I was already caring for patients with cutaneous T-cell lymphoma. And as you know, it was one of our objectives was to find targeted therapies, that is antibodies that could target only the tumor cells and not targeting the reactive tumor T cells. And so I worked with Dr. Bensussan, who is an immunologist, and he had access to many, many antibodies from the world cluster of differentiation, from antibodies targeting natural killer cells. And as you know, natural killer cells express a lot of antigens on their surface, and there is a great variety of these antibodies, killer inhibitory receptors, killer activating receptors, and we looked for the staining of Sézary cells with all these big panel of antibodies, and all were negative one. And by chance, it's not really a chance because it was a systematic review, we observed that patients with cutaneous T-cell lymphomas were, in the majority of cases, 90% of Sézary cells were expressed this new marker, KIR3DL2, that is normally expressed on a minority of NK cells. And this marker was atypically expressed by Sézary cells. And immediately we thought that it would be a nice target to develop antibodies to destroy these tumor T cells and not the normal T lymphocytes. However, there had been a lot of steps after that to control this, to do what is if these antibodies could destroy, really have ADCC, antibody-dependent cytotoxicity. And then we had to pray this that these antibodies were able to destroy the tumor T cells in mice models, also in vivo models of the patients to do the lymphocytes of the patient, the NK cells, and the Caesar cell of the patients to demonstrate that these antibodies were able to kill the cells in that model. And also, finally, in big animals, and the partnership with ANA was extremely important at that step. These new preclinical studies to raise a new panel, new antibodies to select the best antibodies, clinical for finally arriving to the clinical trial and the phase 1 studies, first in human phase 1 studies, and now phase 2 studies. This is quite a long story.
Yeah, and that was a very quick. Maybe how many years was that to get from the lab and that kind of first start to phase 1?
The first paper was in Blood in 2000, so 20 years ago. But it's not so easy to find an industrial partnership because, as you know, there are many other possibilities of trials, and at the beginning it was not a priority of our partner, but finally it became progressively a priority, and and we could go on longer with the clinical part and go into the clinics. Yeah.
Yeah. Wow. So I think that gives everyone a little insight into all the work that goes before we even get to a phase 1. And maybe I can turn to Dr. Porcu at this point to talk a little bit about the phase 1 clinical trial. Maybe we can talk a little bit about what is a phase 1 trial, what are we trying to do? Because as Dr. Bagot just mentioned, this preliminary was all in mouse models in the laboratory and so forth. So we're not testing yet in human beings. So maybe you can talk a little bit about phase 1, how do we get there? What do we do with that? And then maybe some of the outcomes that we learned from the phase 1 clinical trial. And if you can, maybe give us a little timeline. So we get a sense for when did the phase 1 clinical trial begin, and then kind of take us up to the phase 2, and then we'll jump into where we are with the phase 2. That makes sense.
Absolutely. So I think that the description that Dr. Bagot made of the long time required to develop a drug from the laboratory to the clinic, it's unfortunately it's still true, and still takes a lot of time to kind of get things to the clinic. However, the good news is that once we start the earliest phase of the clinical trial development of a drug, then things start to move faster. And to kind of address your question of the timeline, typically phase 1 clinical trials enroll over a period of 18 to 24 months, sometimes a little longer, depending on variable things. For example, the COVID pandemic, just in the past 2.5 years, certainly has put a dent in the timeline for a lot of clinical trials, although not for this one, not for the phase 1. So phase 1 clinical trials are the very earliest phase of development of a new drug. And some of them are, as Dr. Bagot mentioned, first in humans. In other words, that's a drug that we know a lot about already in terms of the biology, kind of the kinetics, the absorption. This is all from preclinical work. But then at some point we have to test the drug in patients. Nowadays, these clinical trials, fortunately, are conducted with enough information about that we have a presumption of efficacy. Because even though the primary endpoint of a phase 1 is really kind of to determine how safe the drug is, and also to look at some of the, how well absorbed it is, how long it stays in the system, and all of that. But ultimately, we really want to make sure the patients actually have benefit from it. This happened with the phase 1 for lacutamab, which was finished in 2020, the drug was effective and the drug was safe. There were 44 patients on the phase 1. In the phase 1 enrolled patients that had both Sézary syndrome and mycosis fungoides. In fact, the majority of the patients on the phase 1 were patients with Sézary syndrome, which, as everyone knows, including on this call here this morning, Sézary syndrome is a rare subtype of cutaneous lymphoma. There's a very high unmet need. There still are no, very good treatments, and certainly there's curative treatment. So the phase 1 was really kind of successful in the sense that it demonstrated safety. The endpoints of phase 1s typically are something called maximum tolerated dose and dose limiting toxicity. These are kind of standard language, in drug development. Ultimately, we want to make sure that by escalating up the dose, that finally reaches a blood level that we know is going to be optimal, perhaps, from the standpoint of the response, or even from the standpoint of the safety and the balance between the two. From that standpoint, really kind of the phase 1 was very successful, and the drug was safe and was effective. There was a significant response rate, particularly in Sézary patients. So it's a 35-36% response rate in patients. So for a phase 1, that's a phenomenally good response.
That's very exciting, and that just finished up in 2020. So again, looking at our timeline from when Dr. Bagot first presented preliminary data in 2000, that's that 20-year window to get through the phase 1 clinical trial. Dr. Bagot, is there anything you'd like to add about what we learned from the phase 1? I think one interesting thing is initially you were targeting Sézary syndrome, but maybe you can speak a little bit about, kind of how mycosis fungoides also got into the game, if you will, but that's not the right way to say it. But adding the mycosis fungoides as a also therapeutic
Interesting.
target.
Yeah. Actually, this included patients with Sézary syndrome, and we were surprised that some patients had a very good and prolonged responses, even with low doses, because it was an escalating dose. And so this was extremely encouraging. And also what's very encouraging, what we have almost no side effects. And so that was very interesting to go on. And this phase 1 allowed to determine the dose and to pave the way for the second phase 2 study, where we'll speak afterwards, and to try to include more patients with mycosis fungoides, because it is extremely important to evaluate whether this drug can also be efficient in patients with mycosis fungoides and not only in patients with Sézary syndrome.
Yeah. And I think that's really great, as we all know, and most patients know, especially those who are dealing with the more aggressive and challenging variants of cutaneous lymphomas. It's hard to, we've got a smattering of treatments, but there isn't anything that we more. Let's just say that. So this is very exciting, and to know that toxicities and safety is really important, because this tends to be a long-term treatment. So unlike other cancers where you may be able to tolerate a higher toxicity of the drug for that short window of time and have effectiveness, in cutaneous lymphomas that's generally not the case, because it's a longer-term treatment, at least at this point. So wow, that's exciting. So here we are in 2022, and I won't say post-COVID, but at least coming out of the restrictions of COVID, and the TELLOMAK trial has moved into phase 2. So perhaps I'll let either of you jump in here to explain a little bit about the phase 2 trial. What are the parameters? Kind of what are we looking for, and where are we today with the phase 2 trial, and with any data that you might be able to share.
Martine, you want to go?
So this TELLOMAK phase 2 trial includes several arms. And so there is a first arm of patients with Sézary syndrome. And this arm is supposed to include 60 patients with a Sézary syndrome. We had several problems to include. Of course, the patients that are supposed to be included in this Sézary arm are Sézary patients having failed at least 2 previous treatments, including mogamulizumab. Because you know that mogamulizumab is now a reference treatment for Sézary patients, and the FDA and the European agency asked to position the new treatment lacutamab in patients having failed mogamulizumab. And so this is it was quite a challenge to include patients in this arm, because during COVID some patients didn't go for the treatment for mogamulizumab. Then we have reimbursement problems in Europe. In many countries, the mogamulizumab until now was not still reimbursed, so it was difficult to prescribe it to patients. And also, mogamulizumab is a nice drug, of course, that works quite well in the blood, not always so. But the patient to be included in this trial have to have an escalation of their Sézary cells in the blood up to thousands. So it's not easy to include in this trial, but finally it was possible, and we hope that in the next month, and we will include the good patient to include in these patients are patients who have failed treatment with mogamulizumab for the Sézary syndrome patients. In case going 2 arms for patients with mycosis fungoides, and the patient included in these 2 arms are patients with the mycosis fungoides having failed at least 2 previous treatments, but not specifically mogamulizumab, so they can have failed 2 other treatments. And these 2 arms correspond to an expression or not of the marker KIR3DL2, quite a low percentage, because positive patients are patients having more than 1% KIR3DL2 cells in their skin or in the blood. And the third arm is patient with negative KIR3 expression. And this preliminary study have showed that patients expressing KIR3DL2 are more advanced patients, are more patients more likely to have pre-Sézary blood involvement, B1, or nodal involvement. So patients expressing KIR3DL2 may be a bit more advanced patients. And the arm 2 has already showed interesting results and demonstrated that it is possible with a treatment with lacutamab to have effect on the skin. Because of course we do not evaluate the blood, because the patient do not have evaluable blood involvement, but we already have significant results showing that an interesting effect can be observed on the skin using the international criteria, the mSWAT, also of course evaluation of other compartments of the blood, of nodes, of viscera. But for these earlier stage patients with mycosis fungoides, the evaluation of the is extremely important. So maybe Pierluigi, you can continue.
Sure. Yeah. So I think the other thing to emphasize is, again, the safety. Now, so far in the phase 2 with a significantly larger number of patients, looks like the safety signal that was observed in the phase 1. So in other words, the impression of safety that came out of the phase 1 is always important to confirm it in the phase 2. And this seems to be on track right now to kind of confirm the safety, because as you mentioned, Susan, patients with both mycosis fungoides and Sézary syndrome, they really are looking for long extended duration of treatment. So the safety cannot be possibly overemphasized as far as kind of having a drug that you can live with. And this seems to be the case. The other, regarding the mogamulizumab, as Martine was mentioning, there were 7 patients that had received mogamulizumab in the phase 1. That's how, we kind of knew there were responses in those patients. So it certainly seemed that having received mogamulizumab did not prevent necessarily somebody from responding to lacutamab, and that makes sense, because lacutamab and mogamulizumab, they target 2 completely different markers on the caesary cells and the mycosis fungoides. Another thing is relevant is that they're to see a good responses in the skin because the vast majority of patients with CTCL, including a lot of patients with Sézary syndrome, they have a significant amount of disease in the skin. And what we are learning now with with drug development and clinical trial in CTCL is that very important to look at what we call response by compartment. So in other words, obviously we want to see how people respond in the skin, and as Martine mentioned, there is this tool called M-SWOT, which is kind of a visual way of calculating the percentage involvement on the body surface, and that's very important. The M-SWOT also takes into account whether people have patch, plaque, or tumor, because the clinical meaning of those different lesions is important. So we also want to keep that into account. Then blood, which is measured by flow cytometry, is a kind of power twist. And then we also want to make sure that people respond in the lymph nodes, and if in situation where the CTCL is also in organs like liver, spleen, lungs, we want to make sure that people respond. That's what's called global response. Now have the answers according to this global score, which is different from clinical trials in the past. So it's very difficult, for example, to try to compare response rates with drugs that are being developed now compared to drugs that had been developed, say, 20 years ago, because the response criteria are completely different. And
that's
they're
really
more
a good point.
Right now. So, yeah,
right.
So in the past, probably the response rates were not inflated, but I mean they were higher because it was less rigorous in the way we're measuring response.
That's very interesting. I think that's something that, from a patient perspective, if you're trying to determine whether you want to participate in a clinical trial or not, and sometimes people look and compare, and I know that always comes up in conversation with regards to response rates, and in cutaneous lymphomas, we tend to see, from a layperson's perspective, what appears to be maybe lower response rates. But I think your point with regards to safety, regards to overall effectiveness and so forth, is really important. And sometimes you have to really look beyond the numbers that might be presented in a study and so forth, because, as you said, it's hard to compare today to a clinical trial data that may have happened 20 years ago. They're much more rigorous. We've learned a lot. We have more data. So it has to be put into context and perspective. And I think that's something really important for patients to keep in mind, especially in our disease, because it's not like we have a plethora of clinical trials and data. I mean, really, when you look at the history, we didn't have any new treatments until, the HDAC inhibitors that came in the mid-2000s, and maybe some of the retinoids for more skin type of disease. And until lately with mogamulizumab and brentuximab, there really hasn't been anything. So the fact that we've now got another agent in phase 2 clinical trial that's showing efficacy and safety, particularly for more challenging and advanced disease, is, I think, very exciting. From the patient perspective, I think it gives people a lot of hope that there's more coming down the line. And I guess one of the other things to mention is, this is an international clinical trial. So obviously, Dr. Bagot, you are in France and conducting the trial, and Dr. Porcu, you're here in the U.S. Do you have an idea of how many sites around the world are participating in the TELLOMAK trial?
In the TELLOMAK, I can remember, I'm remember. I know that there are several many sites both in US and in Europe. I don't know how many are open until now. I would like to say that, for example, this availability of a new antibody is extremely important because we know from the situation in several hematological diseases that they have now a lot of new treatments. If you look at myeloma, they can do sequential treatment, combined treatment, and so for the field of cutaneous T-cell lymphoma, it is extremely important to have new antibodies, this one, and I hope so many other new ones, and to combine, to rotate between the treatments. And it is also extremely important to have a safe new treatment for earlier patients with the earlier stages, patients with mycosis fungoides, with disseminated patches. They have a good prognosis, but sometimes they do not respond to the available treatments, and so they need new treatments, and new treatment that will be well tolerated and extremely safe. And so this is very, very important to develop these new possibilities to treat the patients.
I would agree. Lacutamab essentially is a form of immunotherapy. I don't think we've kind of mentioned that explicitly, but like Martine is mentioning, immunotherapy is really an important piece of cancer treatment in general right now. There are many different forms of immunotherapy. Monoclonal antibodies clearly is one of them. Mogamulizumab also is a monoclonal antibody. So we're learning a lot about ways of doing immunotherapy in different shapes and in different forms. And everything that we do, this is also important for the patients to understand that every advance that we make step by step with studying new targets, new therapies, enrolling in clinical trials, that builds up the foundation for faster and better sort of build up of new treatments down the road. This is really kind of a way that everything is connected. We all learn, and both scientifically and clinically. So participating in a clinical trial, in addition to sort of being potentially good for the patient who participates, and that's why I said at the beginning that people participate in clinical trials because they really sort of want to have something that is going to benefit themselves. That's a primary reason. That's a good reason. But I think that also by participating in clinical trial, one contributes really to the overall advance and growth of knowledge and understanding. So we're kind of all participating in making things moving faster and better. So I think cannot be underestimated.
Yeah, that's a really good point. And before we wrap up, I'll give folks some resources and information on where they can find this particular clinical trials and others that are ongoing in cutaneous lymphoma, because it's important for all of us to participate to help all of those of us that are continuing to live with this disease. And ultimately, put the Lymphoma Foundation out of business when we can cure it and no one needs us anymore. That's the big vision, right? But until then, we really need to continue to do the research and the clinical trials and sharing of data and even post-clinical trial. I think, as you mentioned, Dr. Bagot, the importance of being able to have a lot of options in the toolbox to either combine or before and after, and different avenues of targeting this disease, because it's very smart as well, and learn to evade some of these therapies. And so the more options we have, the better opportunity for a patient to be able to live a good life, even if they happen to have this disease. So I'd like to ask a question. Where do we go from here? If we look in a crystal ball, what's the time from where we are today with the phase 2 clinical trial and going forward? Where do you see the next timeline and the time frame, and what's your vision for what happens from this point forward? Dr. Bagot, do you want to take a shot?
Yes, soon. So the TELLOMAK trial is currently ongoing. Of course, it has been a little slowed down during the COVID period, but now everything is going progressively to normal. And we hope to complete the trial during the next month and to completely finish at the end of 2022 or beginning of 2023. We also have to have a point, so a new update of the evaluation during the second part of 2022. And finally, to have the final results during the year 2023 and to be able to evaluate the different arms of the trial, now the Caesarean, the two mycosis fungoides arms, and to be able to really complete the trial during the next year. It's a big hope.
Wow, that's great. Yeah, that's exciting. I mean, especially given kind of the COVID interruptions that we all, and I know all of your clinics and the labs and everyone has experienced. So that's very, very exciting. And I guess from here, is there anything else that either of you would like to share with the patients with regards to the TELLOMAK trial? I should also say, just to point out, the name of the drug is lacutamab. So we always have fun with naming all of the drugs, and it gets confusing with regards to, before it gets into clinical trial, there's a number, and then then you get a name, and then once it gets approved, then you have a commercial name. So, and we use all of those sometimes simultaneously. So the name of this drug is lacutamab, and the clinical trial name is TELLOMAK, and the company that is hosting the trial is Innate Pharma. So just to kind of level that, because it gets very confusing, I think, for everyone on our side to wrap your head around which is which and who's got what. So is there anything else that either of you would like to share with the patients with regards to this trial, or final words or thoughts on what's next?
So just a couple of comments, final comments here. One is to go back again to the kind of the timing and the timeline of all, and the fact that, as Martine mentioned at the beginning, was very difficult to actually get, a partnership with a pharmaceutical company to explore. That's something that, everybody, like Martine, who's been in the laboratory and trying to kind of, develop new treatments, we all run into it. I think the partnership that Martine was able to establish with Innate Pharma, I think, is incredibly productive. It takes two to tango, so to speak, right? I mean, it's you really need to have somebody who really believes in a new target, in a new drug, and then you have the company that buys, in on that. I think that I see that it's always a very complicated landscape. I see that there are pharmaceutical companies that are interested in rare, not many, but I think that there are, and I think we have to really, those are the ones that I like to work with, because patients with rare blood cancers, CTCL being one of them, but not the only one. I think that that's where we really need to make a big difference. And second is the regulatory agencies. We talked about the FDA. The FDA gave fast drug designation to lacutamab after the phase 1, which means that the U.S. federal government regulatory agency saw a potential for the drug, and that kind of gives you a better way to sort of develop it. The same for the European Union, I believe, sort of gave a very similar sort of route, and this is important because it takes a village, to sort of to really take these drugs to the patients, and is so people work together. I'm optimistic that, like Martine, that the TELLOMAK trial will complete enrollment sort of soon, quickly, and that is going to be it looks very promising. So I think we really hope that the outcomes are going to be favorable, and hopefully we can give this drug to patients routinely soon.
That's great. Dr. Bagot, anything else that you'd like to share?
No, I would like to thank you for this very nice talk all together. We all hope that we will be able to complete the trial very quickly for the benefit of our
Yeah. So thank you both for joining us. Thank you for sharing the background and the data. I think this was a really informative milestone in this particular clinical trial to help patients understand where things are, and really look forward to the next steps and the next opportunity to share data, hopefully from the phase 2, and figure out what's next from there and get this as fast as we can into the clinic in the best possible way, of course, around the world. I think that's one of the other things we're learning too. And, just because we have a new therapy that is available, it doesn't mean that everyone in every country has access to it. So that's another challenge that we're looking at, trying to get ahead of, and hopefully we can do that as things move forward here with the TELLOMAK trial as well. So thank you both for joining us. It's really been a pleasure to have you, and really grateful for all the work that you do and what you continue to do for patients, both in the lab and in the clinic, and in sharing your knowledge with young researchers who are coming into the field. I think that's also so important, so that we can continue this really good work that you all are doing. So thank you very much. Deeply appreciate your time and your valuable insights today, and look forward to next steps as we move forward with the clinical trial. Thank you both.
So, thank you, Susan.
Thank you, Susan. Thanks. It was great to be with you, Martine. So good to see you. And good night, everyone.
Thanks so much. Yes, good night, good afternoon. And before we head off, I just would like to share, if you are interested in learning more about the TELLOMAK trial with lacutamab, you can go out to our website and look at the clinical trials. We just updated and gave you a new way of searching for clinical trials. You can see what sites are open. You can also go out to clinicaltrials.gov, and you can find the available sites that are hosting the trial and see if one is near you. Also, ask your physicians if they're aware of it, if you would be a candidate. All of the information that looks at who would be eligible and so forth, some of that basic information is included on our website, and for sure on the clinicaltrials.gov website. But talk to your physicians to see if you're interested, and if that might be a clinical trial that you are eligible for and interested in pursuing. Because, as I think both of our physicians said, it takes all of us, and hopefully the individuals participating in the trial have a large benefit, but also by participating you help other people as well. So thank you all. It's been a pleasure. I hope you found this informative, and as we get to new milestones for all the clinical trials that we're staying on top of, we'll bring them to you in various different formats. And again, you can find a lot of the information about clinical trials on our website. So I welcome you to check that out, and we update that as much as we can. So, and if you have any questions, you can always call us or send us an email. We're happy to help fill in some of the gaps or answer some questions for you. So thank you, everyone. It's always a pleasure to be with you, and I look forward to seeing you all soon. Take good care.